Hi everyone, and welcome to our video on regulating blood glucose. This is part of module 5 section 4, and what we're going to be looking at today is specification reference 5.1.4d, how blood glucose concentration is regulated. So first of all, we need to understand the kind of levels that blood glucose should be and the issues if it moves beyond those levels. So normal blood glucose concentration should be between 4 and 6 millimoles per decimeter cubed. If your blood glucose falls below 4 millimoles for a long period, then you'd be described as being hypoglycemic. And if it moved above the 6 millimoles for an extended period of time, then you would be hyperglycemic. So hypo is the low, hyper is the high. In terms of why this is an issue, if we think about hypoglycemia, so the low blood glucose, then basically we're not getting enough glucose to our cells. So because of that, we won't be able to carry out the processes we need to, and therefore the first kind of things you're going to see are tiredness and irritability. If this is more severe hypoglycemia, then we're basically starving our cells of the glucose they require to carry out those basic functions, and therefore we're going to see confusion, you're going to have potentially seizures, maybe become unconscious, and in the worst cases actually die. So quite a serious situation. On the other end of the spectrum, when your blood glucose is too high, then that is described as hyperglycemia, and this can lead to organ damage in a range of different organs. So what we're going to see is that if your blood glucose concentration is consistently higher than 7 millimoles per decimeter cubed, then what we would find is you are going to be diagnosed with diabetes mellitus. What should happen then in terms of how our body copes with blood glucose concentration and keeping it within the acceptable range of between 4 and 6 millimoles per decimeter cubed? Well, first of all, we need this monitoring process. So your blood glucose concentration is actually constantly being monitored by the cells in the islets of Langerhans within the pancreas. And if we see a change away from that acceptable concentration, then a hormone is going to be released to then bring that concentration back to the acceptable level. And hopefully we're thinking, oh, that sounds familiar. And it should, because it's negative feedback. Those hormones that are being released are then going to act on the hepatocytes. And if we remember, hepatocytes are liver cells. So you can break it down. Hepato is all to do with the liver, and cyte just means cell. And those hepatocytes, depending on which hormone it is that is then binding with them, will either break down glycogen or create more glycogen. Let's now have a little look at what happens in those two scenarios, blood glucose too high, blood glucose too low. So we'll start with our blood glucose concentration being too high. So if we've got a surplus of glucose within our bloodstream, then the beta cells of our pancreas are going to detect that. So the glucose enters those beta cells, and as a result we produce ATP. When we get that ATP accumulating within those beta cells, the potassium ion channels are going to close. That then means we get an accumulation of the potassium ions and a potential difference generated across the cell surface membrane. As a result of that, the calcium ion channels are going to open, calcium ions then enter the beta cell, and that leads to the exocytosis of the vesicles that contain the insulin. Once the insulin is in the bloodstream, then it's obviously going to travel to those target cells. And we do have the majority of these located in the liver cells and muscle cells, but there are others that also respond to insulin in places like your brain. Now, when the insulin gets those target cells, it will bind to the receptor on the cell surface membrane, and that activates the enzyme tyrosine kinase. And tyrosine kinase just causes phosphorylation of inactive enzymes in the cell, and that causes an enzyme cascade. So what actually happens in that cell? The vesicles containing those transporter proteins for glucose are then going to be fused with the cell surface membrane. So basically what we're now seeing is that we've got more transporter proteins in the cell surface membrane for glucose, which means more glucose from the bloodstream can actually enter those cells. When that glucose enters the cells, there's a variety of things that could happen. One, we can use it in respiration. Secondly, we could convert it into glycogen, and we would then be using that as a storage molecule. And that process is called glycogenesis. Now, glycogenesis is one of these words that people tend to mix up or think is really quite daunting, but the reality is we can break it down into two parts. So if we have a look at the first bit, we can actually just take glycogen, and that clearly just says glycogen. And then we're going to have a bit of an overlap here, but genesis is basically making. So what we're seeing in glycogenesis is the making of glycogen. So we're basically converting glucose into glycogen for storage. We could also see that glucose being converted into fats, and the end result of all of these processes taking place within those liver cells and the muscle cells is that the blood glucose concentration is going to decrease back to those acceptable levels. On the flip side, if we go with a blood glucose concentration that is too low, this time it's detected by the alpha cells in the islets of Langerhans of the pancreas. Now, the alpha cells are going to secrete a different hormone, glucagon. So the glucagon then travels in the bloodstream to the target cells, which are the hepatocytes, so the liver cells. And when it binds to the receptors on the cell surface membrane, it stimulates a G protein, which leads to the activation of adenyl cyclase. And adenyl cyclase converts ATP into CAMP, that's cyclic AMP, and cyclic AMP activates a series of enzyme-controlled reactions inside the cell itself. So in terms of the effects of our glucagon then, we are going to see more fatty acids used in respiration. So we're swapping that respiratory substrate over from glucose to fatty acids instead. We're also going to see glycogen being converted into glucose. And here's another little process that we need to know the name of, glycogenolysis. Okay, so glycogenolysis, and again, if we break it down, we've got glycogen and we've got lysis. And if we remember, lysis just means splitting. So we are splitting the glycogen back into its constituent molecules, glucose. We then have our amino acids and fats being converted into glucose as well, because we've got a lack of glucose. So we're going to convert other substances to replace that glucose that we need. And this is a process called gluconeogenesis. So again, another looking complicated word, but actually we can break it down. So if we take the gluco first of all, that is relating to our glucose. Neo just means new, and genesis is our making. So we're making new glucose, and we're making that glucose from an amino acid and a fat basically. So the end result of these processes, the glycogenolysis, the gluconeogenesis, and the swap in our respiratory substrate, is going to be an increase in that blood glucose concentration. What we're really looking at then in this example of regulating our blood glucose concentration is a negative feedback system. And we've got two hormones involved, glucagon and insulin, and they are antagonistic. They have those opposite effects to each other. So we've got our little flow chart down at the bottom just to summarize everything that is taking place. And what we can see is we start off with our rise in the blood glucose concentration, detected by the beta cells in the islets of Langerhans. They secrete the insulin, which is detected by those receptors on liver and muscle cells. That then leads to the liver and muscle cells removing glucose from the blood through those processes we looked at, and therefore the glucose concentration falls. If, however, we had a fall in blood glucose concentration detected by the alpha cells in the islets of Langerhans, they secrete the glucagon, detected by the liver cells, and then we're going to convert the glycogen to glucose and therefore release it into the blood to bring it back up again. Now, one of the good things that you can do to learn this is start off with a little flowchart like this, print it out, draw it out, whatever works for you, cover up each of those little bits and see if you can get from one side to the other completely without any mistakes. Keep doing it until you've got it, and then once you've got those basics down, go to the extra detail, add in the names of those processes, add in the specific things that are happening in those cells to cause that increase or decrease in the glucose. As always, I hope you found this video helpful to you, and don't forget to subscribe to the channel so you can see when the next video is uploaded, and of course head on over to the website where you can find a range of other resources to help you in your A-level biology studies.